All right. Hi, everyone. Thank you all for coming. My name is Matthew Ming. I'm a PhD candidate at the University of Texas at Austin, and I'm really excited to talk about some of the work that we've been doing recently looking for genome-wide signals of sex differential selection. This work was just uploaded to BioRx like 5 days ago, so I'm really excited to share it with you all. Sexual dimorphism is ubiquitous in nature. It's seen in many sexually reproducing organisms like these lions, peafowl, humans. Specifically in humans, humans are supersexually dimorphic for a lot of traits, including diseases. So understanding the evolutionary mechanisms underlying sexual dimorphism and the mortality load associated with it is vitally important. It's believed that sex differential selection underlies sexual dimorphism. So what is sex differential selection? Sex differential selection, or SDS, is this concept where selection on alleles differs between males and females. So for example, imagine we have this A and T alleles in a population, and the T allele is deleterious in males but beneficial in females, and vice versa for the A allele. Over the course of one generation, survival between males and females will differ, and so viability selection will lead to differential survival between males and females, carrying different alleles, within one generation, leading to a difference in allele frequency between sexes in adulthood. So I adapted this figure from Cole et al. 2024, which is currently in preprint on BioRx, and it's been theorized that sex differential expression, which is where gene expression levels differ between males and females, is related to sex differential selection. So what we wanted to explore is, can we detect a connection between expression and selection, and can we find signals of genome-wide sex differential selection in humans specifically? So people have searched for such signals using a variety of methods, but the results from this research is somewhat contentious. We have sort of two sides in this debate. On one hand, some research has shown evidence that sex differential selection is real, and specifically some papers have given evidence that sex differential expression drives sex differential selection. On the other hand, there's been some work put out that suggests that signals of sex differential selection are just due to statistical artifacts. So today I'm going to zero in on one paper, which argues that sex differential selection is real, and we can detect genome-wide signals of sex differences in expression driving sex differential selection. So this was put out by Changdi Chung and Mark Kirkpatrick in PLOS Genetics in 2016, and these two are co-authors on the work I'm presenting here today as well. So in their paper, they propose a model that relates sex differential selection to sex differential expression, and so they use this between sex FST as a statistic for quantifying between sex allele frequency divergence, and it sort of therefore acts as a measure of the effect of sex differential viability selection. Then they take this between sex FST, and they model it to have a relationship with sex differences in gene expression, and they found that this characteristic sort of Twin Peaks pattern is generated by their model. However, we think that there are some theoretical caveats which haven't been considered up until now, which affect how we interpret the model and the Twin Peaks pattern. So we hypothesize that the pattern is not due to a relationship between sex differential selection and expression, but rather due to statistical artifacts. Okay, so the foundation of the model is in the behavior at low between sex differences in gene expression. So this portion in blue here uses a well-supported theoretical mathematical model, and it suggests that FST has a quadratic relationship with low differences in expression. This portion of the model also makes some important assumptions, a main one being that selection is equal and opposite between the two sexes. So this means that if an allele has a fitness effect of S in males, it's going to have a fitness effect of negative S in females. So overall, we think that this portion of the model is well-supported and it's good. However, the authors don't have a clear mathematical model for the extreme values of sex differences in gene expression, so meaning those genes which are only expressed in males or only expressed in females. So they suggest that FST should go to 0 when the genes are only expressed in one sex. But they use what we believe is some sort of shaky intuition, and they rely on that symmetric selection assumption that I just mentioned, but that assumption was applied to low sex differences in expression. So again, that blue region, and they applied it where it doesn't necessarily work at the extremes. Then the Twin Peaks pattern just sort of comes from an interpolation between that mathematically supported middle part and the extreme unsupported extreme values. And so I showed the interpolation in dotted lines here. So we think that this model is sort of on shaky ground from a theoretical standpoint, but we wanted to know if this model and the Twin Peaks pattern hold up when we test them empirically. So for the following analysis, I want to mention that we followed the original Twin Peaks methodology and datasets. So we used 1,000 genomes, which is a human database, for the allele frequencies and for calculating FST. And then we used the pilot GTEx v3, which is again a human database, and we use that to get sex differences in gene expression. So I should note that in GTEx v3, there are no individual level sex labels, so the analysis was restricted to just the ovaries and the testes, and you can see that they have pretty limited sample sizes. So here's the curve of the original data using the original methodology, and you can see that it has this clear Twin Peaks pattern. So what we're looking at is a curve generated using a 4th degree polynomial regression. And it has between sex FST on the y-axis and sex differences in gene expression on the x-axis. So then to investigate if Twin Peaks is real, we permuted the sex labels in the expression data. So here's some dummy data to demonstrate how we did this. You don't have to pay attention to the numbers because I just made them up. Basically, we randomized which sex labels were assigned to each column of the expression values. And permuting the expression sex labels should break up any of the associations between FST and differences in expression. So if the Twin Peaks pattern is really due to sex differences in expression driving sex differential selection, we should see no Twin Peaks pattern after the permutations. So we're going to permute the sex labels, we'll recalculate the sex differences in expression, we'll rerun the regression, and then we'll do that a bunch of times. And that should give us a null distribution of curves, and the null hypothesis is that there's no relationship between expression and selection. So it turns out that we find that the Twin Peaks pattern is consistent with this null distribution. And you can see the blue line here, which represents the real data, is not an outlier when we compare it to the sex permuted null curves in orange and red. And in fact, we find that over 20% of our sex permuted iterations still have a Twin Peaks shape. And those iterations we marked in red here. So the empirical Twin Peaks pattern is not significantly different from the null distribution, and we often get Twin Peaks patterns even when we permute the sex labels. So based on these results, we think that perhaps Twin Peaks is just due to statistical artifacts, but that doesn't mean that sex differential selection isn't real. So if that's the case, is it possible to find any genome-wide signals of sex differential selection relating to sex differential expression, perhaps by using other methods? So to investigate this, we built upon the Twin Peaks model to get an improved regression method. So this here is the equation for the original model proposed by Cheng and Kirkpatrick in 2016. And so this is the part of the model which was in blue before, which I said was sort of good and mathematically supported. So basically this models how viability selection acting on sex differences in expression could lead to allele frequency differences between the sexes within one generation. So as I previously mentioned, we sort of assume equal and opposite strengths of selection in males and females. And then FST, which is the part in green, is how we'll quantify the sex differences in allele frequencies driven by viability selection. And then the variable delta in the middle is the measurement of sex differential expression, and we weight that by allele frequency 4pq. So if we take FST and we linearly regress it on 4pq delta squared, we get a regression coefficient, and we call that A. And then this A can sort of be thought of as strength of sex differential selection driven by sex differences in expression. So here's a cartoon of what the regression might look like. Each point is a gene, and we'll regress FST on the y-axis against 4pq delta squared on the x-axis, and then the slope of the regression is that A variable. So this is what the regression might look like if sex differential selection is strong and is related to expression. And then here's what we might expect it to look like if selection is weak or it's not related to expression. And then the idea is we're going to perform this regression, we'll get a point estimate for the slope, and then we'll again permute the sex labels, and we'll get a new delta value, and then we'll redo the regression and get new A values, and then that distribution of sex permuted A values will be our null distribution. So we're going to look across many tissues and see if any of them give us a signal of sex differential selection. So for this analysis, we not only updated the method, but we also updated the data sets. So for the next bit, we're using Nomad v3, another public human data set, and this has an entire order of magnitude more samples than 1,000 genomes. We're also using GTEx v8, which has hundreds of individuals, all individually sex labeled, so we can use other tissues besides just the gonad. So we're not constrained in the same way as the original analysis. So even when we use this updated method and the updated data sets, we still see no significant evidence of genome-wide signals, sex differential selection being driven by sex differential expression. So this figure shows a subset of some of the tissues we analyzed. Basically, each row is a tissue. The x-axis is our estimate of that coefficient A, the regression coefficient. And then so that represents the potential strength of sex differential selection. And then the blue dots are the point estimates of A. and the brown bars are the null distribution acceptance regions. So if you compare the blue dots with the brown bars, you can see that we found across none of these tissues, none of them gave a point estimate that was outside of the null distribution acceptance region. And in fact, if you look across all of the 39 tissues that we analyzed, none of them had significant point estimates. So basically, none of the blue dots lie outside of the brown bars, so none of the point estimates are significant. Okay, so why do we not see any signal? For the updated regression analysis that I just discussed, we focused on eQTLs, or expression quantitative trait loci, as sort of the targets of selection. And we did this so that we could assess the effect of selection specifically on the sites which are thought to control changes in expression. However, it's been shown, and I'm specifically going to reference a paper here by Hamanesh Mostafavi and colleagues, which was put out in 2023. It's been shown that there's a bias in eQTL discovery, such that eQTLs under strong selection are less likely to be detected in mapping experiments. So basically, by using eQTLs for detecting this signal, we're probably actually missing out on the sites under the strongest sex differential selection. There's other factors that could also lead to us missing this relationship, such as maybe the relationship between selection and expression is really rare, or maybe it only occurred in the past and is not currently ongoing. If we instead use GWAS hits, we actually do see evidence for sex differential selection genome-wide. So work by my colleagues Carrie Zhu and others, as well as a paper that was just released as a preprint to BioRx by Jared Cole and colleagues. These shows evidence for genome-wide sex differential selection, and they use GWAS hits as the sites of selection as opposed to eQTLs. And to build on that, we actually have some of our own ongoing analysis where we flip the question and ask what's happening at sites which show strong signals of sex differential selection rather than starting with sites associated with changes in expression. And if we do this, we find that there are strong signals of sex differential selection. And interestingly, the strong signal seems especially present in spermatogenesis-related genes. And I'm sorry, because I'm basically going to brush through what's our most exciting result, probably. But we're still sort of making sure that this isn't affected by the statistical artifacts that I mentioned earlier. Okay, so I want to thank my co-author, Changdi Chung, as well as my advisors, Mark Kirkpatrick and Arbel Harpak, as well as my lab mates in the UT Austin community. Thank you all, and if I have time, I'll now answer any questions. Yes?
Have you thought about focusing on the X chromosome and seeing how that might impact sex differential expression?
Right, yeah, so we're actually specifically interested in sex differential selection occurring on the autosomes. We did do some due diligence to make sure that we're not including any mismapped sites that might overlap with either of the sex chromosomes. That's definitely a super interesting avenue of research, but it's I guess it's sort of outside the scope of what we were interested for this particular study, but definitely something that we're interested in looking at in the future. All right, thank you all.
Hi everyone, my name is Zahra Shafiei. I am a master's student working with Dr. Jason Bertram at the University of Western Ontario, Canada. Today I want to talk about the impact of fluctuating selection on genetic diversity when new mutations are expected to be deleterious on average. Well, the first question is why fluctuating selection? To answer that, if we take a look at the environment around us, we can see that it's not constant. Basically, environment surrounds us, and the real-world conditions are continuously changing. And these changes are due to different factors, such as climate changes, availability of different types of resources, and so many factors that we can name them together. On the other hand, if we consider a population that is experiencing different types of fluctuations, it is expected to see a wider range of gene diversities in that population. However, this is an area there's a lot of unanswered questions and debates about the relationship between selective fluctuation and genetic diversity, which is not answered yet. On the other hand, if we take a look at the previous studies in this area, we can see that most of them are incorporating the assumption of balancing selection. However, I want to emphasize that in our model, we don't have any type of balancing selection. This essentially means that mutations are allowed to go always to fixation or loss, depends on their dynamics. If we take a look at the simulations that we have done in the absence of balancing selection, we can see if we move from a stationary environment to a fluctuating one, trajectories of allele frequencies are going to higher frequencies, which means that there's a change in genetic diversity. Another group of previous works which did not cooperate balancing selection, such as Herta Sanchez on 2008 or Takahata and Kimura on 1979, predicted that selective fluctuation actually reduces genetic diversity. And the justification behind that is selective fluctuation is an stochastic force. So we kind of expect it to behave like genetic drift, which is the essence of adding some noises in our model, and that pushes our mutations to fixation or loss. However, in most of these models, we see the footprint of one more important assumption, which is that the distribution of selection coefficients are symmetrical, meaning that in those types of models, they considered deleterious mutation are likely to occur are as likely to occur as beneficial ones. We know that in real world, this is not a correct assumption. We know that new mutations are on average deleterious, and it's because that the distribution of the fitness effects has a shift toward negative, toward deleterious mutation. In this study, we modeled selective fluctuation considering a haploid population of n individuals with two possible alleles A and A, with the fitness of 1 plus sigma and 1 plus tau, where sigma and tau are two independent random variables. And they are independent from each other in one generation and across generations. These variables have an identical variance called V. Now, we want to take one step further and study this assumption that says new mutations are expected to be deleterious on average alongside with selective fluctuation, meaning that we are modeling the bias toward deleterious mutation with a parameter delta, and we call it the negative shift, and delta in this model is the expectation of sigma minus tau to be minus delta. Now, it's time to quantify genetic variation. As we know, there's a lot of methods for doing this, and in this study, we are using the average heterozygosity, which is the following formula, where Vg is the genetic variation or diversity, and X is the frequency of allele A. Let's unpack this equation a little bit together. If I consider fix as a fixed value and go to my population, randomly pick two individuals and come out, there is 2 times x1 minus x is the probability that one of those individuals carries allele A and the other one carries allele small a. That factor 2 there shows that there is no inherent difference between these two alleles. Basically, they both have the same probability of being picked. On the other hand, we are seeing an expectation there showing that we are taking an expectation over an infinite number of replicates in our population. The reason behind that is the frequency x is actually not a fixed value. It varies with time, and we need to account the effect of all of different values of x and account for their different probabilities. Now, with that in mind, we used the previous formula and we calculated genetic diversity for each pair of delta and V, which is the negative shift and selective fluctuation, and this led us to have this heat map. There's a lot to talk in this heat map. However, between all of those, I want to mention and emphasize on two important factors. The first one is, in the absence of selective fluctuation, meaning that when V is almost equals to 0, if I start increasing the negative shift delta, I can see a reduction in genetic variation. However, as soon as I add a little bit of selective fluctuation on the rightmost corner of my plot, I can see that genetic diversity doubles. This essentially shows that in presence of the negative shift, selective fluctuation actually increases genetic diversity. Well, what does it exactly mean? It means that if I consider my environment having a bias, let's say delta equals to 0.003, and then add fluctuation to that environment, this is what I'm going to see. the trajectories of allele frequency can reach very high frequency in comparison with the time that I don't have any type of fluctuation. Well, the next question is how strong my fluctuation needs to be in order to see this pattern. Or if I pick a good range of values for my fluctuation, is this range comparable with the range that I see in the nature? To answer these questions, we used the diffusion approximation and we derived the equation of V equals to delta. Well, V equals to delta is the point at which the influence of selective fluctuation is comparable with the influence of the negative shift on the allele frequency. Now, let's take a step back and again go to our heat map. You have seen this plot, right? Let's zoom out the bottom part where V and delta are equal to each other. If I zoom in, you can see the second panel at which the red line separates the region at which Vg has high value from the region that Vg has lower value in the darker shades. In other words, if I want the selective fluctuation to matter, I need to consider V bigger than delta, meaning that I always need to stay at the upper triangle, because if I'm down, the negative shift will kill off all of the variations. Well, it's good. It's a good conclusion, but still we are looking at parameter range for this comparison. Can we answer that question with this equation? Well, maybe yes, maybe not. Let's take a look at the definition of our parameter. Well, we know that V had the scale of a selection coefficient variance, because basically V was the variance of two parameters that described my selective fluctuation sigma and delta. So the expectation of sigma minus delta squared is equals to 2V. On the other hand of my equation, I have delta, which has the scale of a typical selective fluctuation, with the equation of expectation of sigma minus delta equals to minus delta. So I can see that the scales are different in this equation, meaning that in order to have the comparison for the range of my parameter, I need one more step. And that step is considering the standard deviation, denoted with a square root of V, instead of the variance. Now, all in all, we are trying to say that from heat map, selective fluctuation matters if we are in the regime where V is bigger or equal to delta. And in order to complete this comparison and have the right scale for our comparison, we need to use the standard deviation. Since the values of selective fluctuation in this model are small, very smaller than 1, we will have this inequality, as you can see in this slide. Let me remind you that delta is the selection coefficient due to the negative shift, and on the other side, selection coefficient due to the fluctuation is the square root of V. So I have these two components, and as a numerical example, if I pick the value 0.1 for my V, the square root of that is going to be 0.3, which is the size of my selection coefficient. To sum up, we started with the model of selective fluctuation with two independent random variables, sigma and delta, and we added the assumption of the negative shift toward deleterious mutation to this model. We saw that in presence of this negative shift, selective fluctuations actually increases genetic diversity, meaning that it allows new mutation to reach higher frequencies. Also, we have seen that in order to see this result, we need a very strong selective fluctuation. In other words, high selection coefficient per generation are necessary. Well, why this is necessary? Because as soon as you start thinking about two independent random variables, you are not allowing them to accumulate during the time. So as the next step in our future work, we are planning to add autocorrelation between these two parameters and also consider the negative shift in our model. This will allow our fluctuation to have more time to accumulate, and frequency changes goes upper and over generations in a consistent direction. Thank you all.
Do these patterns
Well, in our model, mutation rate was a constant. Therefore, I cannot, this is an area that we didn't explore yet, but it's a very great question.
In the simulation, like towards the beginning, did you set any parameters around like the criteria of like a standard migration or like criteria of like outcrossing or setting like inbreeding to zero or something?
Well, no, but this is a part of my thesis where we are incorporating genetic drift to this model as well. So basically, the idea is studying selective fluctuation, mutations, the effect of the negative shift, and genetic drift altogether.
So this could be like applicable to say like a non-albino population or a non-mutant population.
Exactly, yes. No worries. Yes.
Thanks a lot. So the heat map you're showing is diagonal.
Mhm.
If you were to zoom in to the diagonal, would you still have the same color shape? Or zooming in and out, would you see the same thing?
Well, no, not exactly. Let me bring this slide and show it to you. The plot that you're seeing on the forest part, that incorporates a wider range of selective fluctuation, right? The pattern on the bottom part is the same. We just zoomed in. We didn't change any assumption. But there's a lot of other factors and equations that we try to use in order to describe the forest panel in the thesis.
Is the assertion that when you don't have a negative shift, the fluctuations reduced genetic variance still help in the simulation?
Yes, it is. So if you take a look at the upper triangle of the forest heat map, so, as much as I'm increasing V, I can see that genetic variation is reducing. So it actually confirms the previous study, also adds the assumption of that. Thank you all.